A bone fragment, the size of a coin, pulled from the dirt of a Siberian cave, sitting in a tray next to 2,000 other fragments nobody could identify. For 4 years, it was just another piece, a scrap of skeleton from a place that had given the world plenty of those. And then somebody ran a protein test, and the scrap stopped being a scrap. It was a girl, 13 years old. She had died inside that cave more than 50,000 years ago, and the genome locked inside her marrow contained something that, by every rule we had ever written about Neanderthal women, should not have been possible. Her mother was one species, her father was another. We will get to her, but first you have to see what nobody saw coming. For most of the 20th century, the picture of a Neanderthal woman was settled. You can find her in any natural history museum diorama painted before the year 2000. She is short, broad, hunched in the back of a cave, holding an infant in one arm and a scraping stone in the other. The men are outside hunting. She is inside, waiting. Local, quiet, brutish, a side character in her own species. The whole consensus was built on almost nothing, 6 partial mitochondrial sequences and a guess that Neanderthal communities were patrilocal, with women marrying in from the next valley over. That guess made it into the textbooks anyway. Neanderthal women were caregivers, gatherers, the static centre of small communities, clinging to a brutal landscape. Everything strange in Neanderthal society, the burials, the symbols, the tools, was assumed to be the work of the men. Ancient DNA did not whisper that picture was wrong. It broke it open. What came out of the Altai Mountains in the last decade rewrote the whole architecture of Neanderthal society. The women had been moving, not a valley over, across continents, for tens of thousands of years. The architecture of their world was not the cave, it was the road. And the proof was sitting in that fragment of bone. To find the first crack, you have to walk into a different cave, about 100 kilometers from where the bone girl was found, in the same range of mountains. There is a place called Chagaskaya. For years it was just one more Middle Paleolithic site. Interesting, but not extraordinary. Then a team, led by the Max Planck Institute in Leipzig, started pulling out something nobody had expected. 13 Neanderthals, from a single cave. Their bones did not come from layers spread across millennia like most fossil deposits. They came from a single, narrow window of time, possibly the same generation, possibly people who had known each other. For the first time in the history of Neanderthal genetics, scientists were not looking at scattered fragments of a species spread across continents and ages. They were looking at a community. The results came out in 2022 in the journal Nature, and inside the genetic data, 2 relationships pulled the entire picture into focus. The first was a father and his daughter. The second was a pair of cousins, people who had lived together, people who had died inside the same cave. The estimated size of the community, about 20 individuals. The effective breeding population, 10 to 20, smaller than a school class. These were not tribes, these were almost households. And inside that household-sized community, the researchers found something strange. They looked at 2 kinds of DNA separately. Mitochondrial DNA, passed only from mothers to children. The Y chromosome, passed only from fathers to sons. Both should tell similar stories if the group had been stable. In Chagarskaya, they told opposite stories. The Y chromosomes were almost identical. The mitochondrial sequences were wildly diverse. The fathers, the sons, the grandfathers had stayed put for generations. The mothers, the daughters, the grandmothers had come from everywhere else. The numbers were brutal. The average coalescence time of the Y chromosome, the genetic clock measuring how recently the men shared a common ancestor, was 446 years. The same number for the mitochondrial DNA was 4,348, almost 10 times older. The simulations the team ran told them the only scenario that fit the data. Between 60 and 100% of the women in that community had been born somewhere else. The men had stayed. The women had walked in. And once you saw the pattern in Chagarskaya, you could go back to the cave with the bone girl and start asking a different question. Who then was her mother? Because mitochondrial DNA passes only from mother to child, the bone girl's mtDNA was a direct reading of her maternal line. The team sequenced it. It came back Neanderthal. That was expected. What was not expected was where the lineage pointed. 2 high-coverage Neanderthal genomes existed at the time. One had been pulled from the same Siberian cave, an older individual called the Altai Neanderthal. Local, ancient, 120,000 years old. The other had come from a cave in Croatia called Vindija. European, much later, about 50,000 years old. The bone girl's mother should have matched the local one. Same cave, same mountain range, same neighborhood, geologically speaking. She did not. She matched the European one. 12.4% of her mother's derived alleles aligned with the Altai. 19.6% aligned with Vindija, a Neanderthal who had lived in Croatia, 5,000 kilometers to the west. The math is hard to absorb on first hearing, so let it land. A girl found in southern Siberia about 90,000 years ago was the daughter of a woman whose ancestral population was more European than Siberian. Either that woman walked there, or her mother did, or her grandmother. Across half a continent, through Ice Age forest and grassland and mountain pass, for reasons no bone will ever tell us. Pregnant or not pregnant, pursued or chosen, alone or in some larger flow we cannot reconstruct. She made the journey. She ended in a cave full of strangers, and she gave birth to a daughter who carried her mother's lineage forward into a place her mother's people had never lived. That was not a one-off. That was the pattern. Chagarskaya proved the structure. Women moving between groups was the rule. The bone girl proved the scale. Those movements could span continents. To understand exactly what her father carried in his genome and why the rest of the picture is about to get worse, you have to look at the most overlooked single sentence in the original paper. Most people scroll past stories like this without ever asking what the bones actually said. You are still here. That puts you in a small group, the ones who want the evidence, not the summary. This is the kind of work this channel exists to do. Subscribe. There is another one coming. The father's DNA had a problem of its own. The bone girl was identified as half Denisovan on the father's side. Denisovans are another extinct human species, sister to Neanderthals, separated from them more than 390,000 years ago. Cousins, distantly. The kind of cousins who had not seen each other in 3,000 human generations. So her father's genome should have been clean Denisovan. It was not. Hidden inside it were 5 long stretches, each about 1 million base pairs in length, that were entirely Neanderthal. Not from the bone girl's mother, from an older event. An ancestor of the father had been part Neanderthal too. The estimate? Between 300 and 600 generations back. 300 generations is roughly 6,000 years. So in this single cave, in this single fragment of bone, there was direct evidence of at least 2 separate hybridization events between Neanderthals and Denisovans, 6,000 years apart. The bone girl was not a freak accident. She was a recurrence. And then the statistical weight of the discovery hit. Of the 6 individuals from that Siberian cave whose nuclear DNA had ever been recovered, 2 showed evidence of cross-species mixing. 1/3 of the sample, from a population that had supposedly been separated for almost 400,000 years. Either the Bone Girl's family was the luckiest find in the history of paleogenetics, or, and this is what the team concluded, the mixing was systematic. Whenever Neanderthal and Denisovan groups met, they bred. Not rarely, not by accident, habitually. The only reason they stayed genetically distinct for so long was that their territories barely overlapped. When the overlap happened, the wall fell every time. And the women were the ones crossing it. The pattern was not confined to Siberia. While the team in Leipzig was sequencing the Bone Girl, another team was working in a cave at the opposite end of Eurasia, in the cold limestone karst of northern Spain. The site is called El Cedrón. The remains there are about 49,000 years old. 12 Neanderthals, found together in a deep gallery, killed and partially consumed in what looks like a single catastrophic episode. When the team led by Carles Laloesa-Fox sequenced their mitochondrial DNA, the result was familiar, even though nobody had named the pattern yet. 3 adult males in the group all shared the same maternal lineage, brothers or close cousins. 3 adult females in the same group had 3 different maternal lineages, strangers to each other and strangers to the men. The same female-biased dispersal that Chagiskaya would later confirm with 13 genomes was already there in Spain in 12. The El Cedrón women had carried something else with them. The team analysed the calculus, the hardened plaque on the teeth, and recovered traces of plants, not food plants, yarrow, chamomile. Bitter, low nutrition, useless as nourishment. The only reason a hominin chews yarrow is to take down inflammation. The only reason to chew chamomile is to settle a stomach. They were medicine. A woman walking from her birth community into a community of strangers was not arriving empty-handed. She was bringing her body. She was bringing her mitochondrial line, and she was bringing pharmacology, knowledge about which plants in the forest would dull pain, ease a fever, bring down a swollen joint. Small, travelling repositories of medical knowledge moving across a continent on foot, one community at a time. Whatever the Bone Girl's mother carried into Siberia, it would have been more than her DNA. Picture it. A young woman, alone or in a small group, walking east through forest she has never seen. The same kind of walk the Bone Girl's mother made, generations later, into a cave full of strangers in the Altai. Within a season, the children of that community begin to recover faster from things their mothers had not known how to treat. Stop and let that image sit. The DNA does not just describe a migration. It describes a transmission. There is one last site you have to see. In the northern Caucasus, on the edge of the Russian forest, there is a cave called Mesmyskaya. It contained the bones of a Neanderthal infant, a girl, 1.5 months old, who had died about 70,000 years ago. Her mitochondrial DNA was sequenced in the year 2000, the first such reading from this end of Eurasia. It did not match the European Neanderthal lineages that researchers had assumed would dominate the entire range. It belonged to a different branch. The map of Neanderthal motherlines was wider than anyone had drawn. Decades later, in 2020, a separate team sequenced the Y chromosome of an adult male from the same cave. That Y chromosome was not most similar to its geographic neighbours. It was most similar to El Cedrón, to Spain. 6,000 kilometres across the breadth of Europe and back into the Caucasus, the male Neanderthal lineages were also flowing. But they were flowing because the women had moved them there generations earlier, born sons in new groups, and the sons had inherited Y chromosomes that traced to places their mothers had walked from. The mothers were the engineers of the entire genetic atlas. Now the final inversion, the one that sounded, when the paper came out, like a misprint. In 2020, the team at the Max Planck Institute sequenced the Y chromosomes of 3 late Neanderthals from Belgium, the Caucasus, and Spain. The Y chromosome is the one piece of the genome that should be unambiguous. It passes father to son, generation after generation, with almost no recombination. If you want to know what the Neanderthal male lineage looked like, the Y is where you check. When those 3 Y chromosomes were placed on the phylogenetic tree of all known hominins, they did not cluster with the Denisovan Y chromosome, their nearest cousin, the way the rest of the Neanderthal genome did. They clustered with the Y chromosome of modern humans. Homo sapiens. The math said the Neanderthal Y chromosome those 3 men carried had diverged from the modern human Y chromosome only about 370,000 years ago, long after Neanderthals and Denisovans had split, long after Neanderthals should have had a Y of their own. The conclusion was inescapable. Sometime between 370,000 and 100,000 years ago, the original Neanderthal Y chromosome had been replaced by a Y chromosome closely related to modern humans across the entire Neanderthal population, in every late Neanderthal whose Y has ever been sequenced. The only way that happens is repeated, sustained interbreeding. Sapiens men mating with Neanderthal women, the resulting sons carrying Sapiens Y chromosomes, those sons reproducing inside the Neanderthal community, and after enough generations, the original Neanderthal Y simply ceased to exist. It was outbred, replaced, quietly, over hundreds of thousands of years. Which means the encounter between modern humans and Neanderthals was not a brief contact event 60,000 years ago when sapiens left Africa. The encounters had been happening for 270,000 years before that, slowly, repeatedly, in places we have not even identified yet, and in every one of them, the conduit was a woman. Now we can come back to her, the bone girl, Denisova 11. The fragment in the tray. She was 13 years old when she died. We know that from the thickness of her cortical bone. We know she was female because her X chromosome appeared at the same coverage as her autosomes. We know she lived about 90,000 years ago. Her mother was Neanderthal, and her mother had walked from an ancestral population closer to Croatia than to Siberia. Her father was Denisovan, and her father carried deep in his own genome the marks of a Neanderthal ancestor from 6,000 years before he was born. She was the daughter of 2 species that on paper had been separated for 400,000 years. In her cells, those species met for the first time in person. She was a first-generation hybrid, not the descendant of a long mixed population, the direct offspring, the single individual in whom 2 extinct human lineages came together and produced a child. Her heterozygosity, the measure of how different her 2 sets of chromosomes were from each other, was more than 4 times higher than any other Neanderthal or Denisovan ever sequenced. She was, genetically, almost 2 people at once. Everything that came before her in this story converges on her. The community of 20 in Chagarskaya, the medicinal plants in Spain, the mother whose ancestral line traced to Europe, the sapiens Y chromosome that should not have been inside a Neanderthal male. All of it was the architecture that made her possible. And she existed inside a single bone fragment, the size of a coin. The implication of finding her is what should break the consensus permanently. Of the 6 individuals from her cave whose nuclear DNA has ever been recovered, 2 showed evidence of recent mixing. Of the 3 modern humans known to have lived during the Neanderthal era, 1, a man named Oase 1, found in a Romanian cave, had a Neanderthal ancestor 4 to 6 generations back, about 120 years before his lifetime. The samples are tiny. The hits are not. Hybrids and recent hybrid descendants keep appearing in almost every meaningful sample we recover. Statistically, that only happens if the mixing was the norm. The women were not exceptions. They were the architecture. There is one part of the story we have not gotten to yet, and it is the part the title promised. What the ancient DNA finally revealed. In 2020, two researchers at the Max Planck Institute, Hugo Zierberg and Svante Paabo, published a study on a single variant of the progesterone receptor gene. Progesterone is the hormone that sustains a pregnancy. Its receptor is the protein that lets the body listen to that hormone. They were comparing modern European women to ancient genomes, and they found that one specific version of the gene, the one that produces more receptors, the one that makes the uterus more sensitive to progesterone, the one associated with fewer early pregnancy bleedings and fewer miscarriages, came directly from Neanderthals. It is not rare. Approximately 1 in 3 European women alive today carries the Neanderthal variant of that gene. They were not given that variant by Neanderthal men. They were given it by Neanderthal women, generations and generations back, whose daughters and granddaughters carried it forward into the modern population by being the bridge between species. That is the truth the ancient DNA revealed. The Neanderthal women did not vanish. They did not die out in a cave in Gibraltar 40,000 years ago, as the popular story still has it. Pieces of them are still walking around. They are inside the bodies of roughly 1/3 of the women in this hemisphere. And one of the things they are doing right now is helping the children of those women hold on through the first dangerous weeks of pregnancy. The species ended, the lineage did not. The women who had walked across continents for 200,000 years did one last thing on their way out. They put a piece of themselves inside the species that replaced them. And inside that species, that piece is still keeping babies alive. Whatever the bone girl's mother gave her, and whatever her mother's mother had carried before that, is part of why 1 in 3 of those women are still here to be pregnant at all. A bone fragment. The size of a coin, pulled from the dirt of a Siberian cave. We thought it was just a fragment. It turned out to be a passport, a girl 13 years old, mother on one side of a vanished line, father on the other, alive in the only generation in which that crossing could have happened. The Neanderthal women were never who we thought they were. They were not silent. They were not local. They were not waiting. They were the road the genome travelled on. If you want to keep following this, what ancient DNA is doing to the story we thought we knew, subscribe. The people who watch these all the way through are the people who already knew the textbook version was incomplete. Turn the bell on if you don't want to wait to find out what the next piece of bone is going to say. Every week there is another piece of the record that turns out to be more complicated and more interesting than the version we were taught.